Welcome to our video lecture on topic C1.3 photosynthesis. This is our additional higher level content. Our guiding questions: how is energy from sunlight absorbed and used in photosynthesis? We're going to talk about so many details here. And then just a little bit about how abiotic factors interact with photosynthesis. Our objectives: we're going to review the basics of photosynthesis. We're going to talk about the functions of photosystems, and those would include oxygen production, ATP synthesis, and NADP reduction. We're also going to look at the Calvin cycle and wrap up with a little look at the interdependence of the light-dependent and light-independent reactions of photosynthesis. Photosynthesis in plants occurs in these amazing organelles, the chloroplasts, which we think evolved from photosynthetic bacteria. Remember that beautiful endosymbiotic theory. Quick review of some of the structures of chloroplasts. They do have double membranes, outer membrane, inner membrane, and then within that inner membrane, there are more membranes, the thylakoids. They look like these stacks of pancakes. Each one of those stacks is called a granum. Grana is plural. Here in the micrograph, we can see all those thylakoid membrane stacks in this granum. There are also membranes that connect the grana. Those are called lamella. We see some lamella here, we see some lamella there. Outside the thylakoid membranes is the stroma. The stroma is here. This is where the light-independent reactions are going to occur. Light-dependent reactions happen in the thylakoid. Light-independent reactions happen in the stroma, still in the chloroplast, but outside those thylakoid membranes. Lots of interdependence between those light-dependent reactions and the light-independent reactions. The overall equation that we need to be able to write for photosynthesis is that we're going to have some carbon dioxide molecules, 6 of them in fact, reacting with water molecules, 12 of them all together. That is going to produce some glucose, C6H12O6, and some oxygen gas molecules, and interestingly, we're going to reform some water. So we're going to break 12 water molecules and then reform 6 water molecules. The carbon dioxide is involved only in the light-independent reactions. The water we need only for the light-dependent reactions. This glucose is going to be produced in the light-independent reactions. The oxygen gas is produced in the light-dependent reactions. We're going to start with lots of details about those light-dependent reactions now. Let's zoom in on a thylakoid membrane. Those thylakoid membranes are phospholipid bilayers, just like our cell membranes. Embedded in the thylakoid membranes are all of these super amazing molecules. We are going to start by talking about PS2 and PS1. PS stands for photosystem. These photosystems are light harvesting complexes. Sometimes we even call them light harvesting complexes. They are composed of these massive arrays of pigment molecules and protein molecules. These pigment molecules are going to absorb light energy, and that's going to cause electrons to get excited. Remember from chemistry, when we did quantum mechanics and our amazing flame test lab, we saw lots of pretty colors of light. The reason that we saw the pretty colors of light in our flame test lab is because our Bunsen burners were causing electrons to absorb energy that caused those electrons to jump to a higher energy level. That is known as an excited electron. This is not a stable configuration for those electrons though, and so they will fall back down to lower energy levels, and we can sometimes see the release of that energy as fluorescence, which is just that release of light energy. We can calculate that with the amazing formula: energy is equal to Planck's constant times frequency. In our photosystems, instead of that energy being released as fluorescence, these excited electrons are wiggly and jiggly, and they are going to pass that energy around these arrays of photosystems. So in the photosystem, we have all these different pigment molecules. One of them is going to get zapped by some light energy. It's now wiggly and jiggly, and it's going to pass that wiggly jiggly energy to the next guy, to the next guy, to the next guy, until we eventually end up at what we're going to call the reaction center. This is the core complex, this reaction center, these antenna complexes, these chlorophyll molecules that are absorbing the light energy. Sometimes we'll also add in some accessory pigments. The benefit of having these accessory pigments like carotene or xanthophyll is that they can absorb different wavelengths of light. So these photosystems can very efficiently harvest lots and lots of different energies of light to carry out lots and lots of photosynthesis. Here is another look at Photosystem II. Photosystem II. We discovered it second, but we talk about it first in the story of photosynthesis. We have these chlorophyll molecules. Here is Planck's constant times frequency. This is the energy from light that is being absorbed by electrons in these chlorophyll molecules. The chlorophyll molecules' electrons get excited. They're going to wiggle and jiggle a little bit extra because they have all that extra energy. And then they're going to wiggle and jiggle and pass that energy onto another, and then another, and then another chlorophyll molecule until eventually that excitement, that wiggly jiggly energy, gets to P680, which is the reaction center of this photosystem number 2. There are 2 special chlorophyll molecules here. We call them P680 because their peak absorbance, the wavelength of light that they best absorb, happens to have a wavelength of 680 nanometers. So this guy is P680 because that's the wavelength of light, electromagnetic spectrum energy, that they best absorb, that they get most excited by. These photosystem II reaction centers, they're going to then start passing electrons off to an electron carrier that we're going to talk about in a couple more slides. When these guys, these P680 chlorophyll molecules, lose electrons, they've been oxidized. Remember that the loss of electrons is oxidation. So when they are oxidized, they are very unstable. We need to replace electrons. That happens with the splitting of water molecules, also using light energy. This is known as photolysis. So photolysis, splitting with light energy water molecules. So 2 water molecules are going to bind to what is known as the oxygen evolving complex. Oxygen evolving complex of photosystem II. There's some manganese and there's some calcium. There are also some oxygen atoms bound in here. They are going to rip apart those water molecules, and the production, what is produced, is some oxygen gas that is released as a waste product. We're going to have some hydrogen ions, also known as protons. These guys are going to stay in the lumen of the thylakoid pancakes. We're also going to have some electrons being released. Those electrons are going to replace the electrons that were lost by P680 when those electrons were passed along to our electron carrier molecule. What's pretty cool about that propagation of oxygen in photosynthesis is that it changed Earth's atmosphere. A long time ago, before photosynthesis had evolved, there was no oxygen in the atmosphere. There were no oxygen gas bubbles floating around in the oceans. And then this super cool mutation happened, and some bacteria, we call them cyanobacteria now, these bacteria started carrying out photosynthesis and producing oxygen gas. At first, that oxygen gas was absorbed in oceans. It bound to minerals that were dissolved in the ocean. Those minerals with the oxygen then kind of settled to the ocean sea floor and became some rock. And then once we had kind of a saturated ocean oxygen level, oxygen gas started to diffuse out of the water, but there were more minerals on the surface of the land, and so those bound to the oxygen, and we had more minerals, more rocks being produced. Eventually, the surface of Earth was saturated with oxygen, and then oxygen gas started to actually enter the atmosphere. And we had this increase in oxygen gas. And because there weren't any humans around with technology able to measure how much oxygen was there, we make a lot of guesses. And so these lines are just showing how the most we think could have been there, the least we think could have been there. It's going to be somewhere in between, right? So what's extra amazing about this accumulation of oxygen in the atmosphere is that it led to our first mass extinction. So we had all these archaebacteria that were super happy in the anaerobic environment of Earth, until this oxygen started to accumulate, and then they died. So our first mass extinction happened because some bacteria started photosynthesizing, and that killed off a bunch of other bacteria. Crazy sauce. Back to this idea of light harvesting in our photosystems. So again, we're going to have these photons of light energy bumping into chlorophyll molecules. That light is going to be absorbed by the electrons. They wiggle and jiggle. That wiggle jiggle happens to bump into another chlorophyll molecule. They wiggle jiggle. And that energy just keeps getting transferred from one chlorophyll molecule to the next. Those pigments that are in the array around the reaction center, eventually the two chlorophyll molecules in the reaction center, we're going to call them P680 in Photosystem II. These guys are going to wiggle and jiggle so much that they actually lose some electrons. And those electrons are going to be donated off to a primary electron acceptor. And we're going to call that guy PQ or plastoquinone. We'll talk about that a little bit more in a couple slides. All of this energy coming from the light that's bumping into these electrons, it's getting passed to these electrons, it's getting passed to our electron carrier molecules. Eventually that energy is going to be bound in the chemical bonds of glucose. So light harvesting is all about collecting energy from the light so that we can carry out some chemical reactions, so that we can store energy in the chemical bonds of those glucose molecules. And again, we're going to talk about chlorophyll the most, but there are other pigments that absorb other colors of light, other wavelengths of light, which is a benefit. This benefit of having pigments that can absorb different wavelengths of light is related to photosystem structures as arrays. So arrays are just these systems of many components that are structured, arranged just so that they can work together to carry out a job very efficiently. So our photosystem arrays are going to be all these different pigment molecules, lots of chlorophylls, but maybe also some of those accessory pigments like, again, carotene. They're going to absorb energy in lots of different wavelengths. Different pigments are specific to different wavelengths. So if we have a variety of pigments, we can absorb a variety of wavelengths. Sunlight is sending white light. That white light includes all the colors of the visible spectrum, so all those different wavelengths. Lots of energy being absorbed from the sun by these different pigments. Also, having this array means that those chlorophyll molecules and the carotene and the xanthophyll molecules are spread out a lot because the sun is sending lots of diffuse energy. It's not like one beam, it's all the beams all over the place. And so those photons are quite scattered as they hit the leaf. Having these many arrays means that there's more of a chance of those photons actually running into electrons in these chlorophyll molecules. The last advantage of having these arrays, because they are arranged near each other, when one chlorophyll molecule electron is excited, it's going to wiggle and jiggle and bump into the next guy and bump into the next guy and bump into the next guy until we get to that P680 reaction center. And remember, again, when P680 loses electrons, we're going to replace them with electrons from photolysis, that splitting of water. So what even happens to those electrons that are lost by Photosystem II and the P680 reaction center? Remember, again, that light is going to bump into some chlorophyll molecules, exciting their electrons. They're going to wiggle and jiggle against each other until they wiggle and jiggle against P680. P680 chlorophyll molecules are then going to lose some electrons. Those electrons are passed to an electron carrier. That electron carrier is called PQ or plastoquinone. When PQ gains electrons, and it's also going to gain a couple hydrogen ions, it is now the reduced form. Remember that losing electrons is oxidation, gaining electrons and also protons is reduction. So the reduced form is PQH2 or plastoquinol. So this PQ, plastoquinone, gains some electrons. It becomes plastoquinol. It's going to pass those high energy electrons that it got from P680 over to this cytochrome complex. These are all parts of the electron transport chain embedded in the thylakoid membranes of the chloroplast. Remember that because P680 lost some electrons, it needs some more electrons, and so it's going to regain electrons from water molecules, that photolysis of water and the oxygen evolving complex of PS2. So now these electrons that are being carried by now the reduced form PQH2 pass to cytochrome. The cytochrome's job is to use the energy from those electrons that have been excited. We're going to use that energy to pump protons, hydrogen ions, into the lumen of the thylakoid. So we're going to take these hydrogen ions that are here, and we're going to pump them into the lumen of the thylakoid. That is going to lead to a proton gradient. We're going to have a high concentration of these hydrogen ions in the lumen of the thylakoid. This is not stable. The universe always tends toward things being at equilibrium, so it would be more stable for us to have equal concentrations of hydrogens inside and outside, inside the lumen and then also in the stroma. However, because these hydrogen ions have a positive charge and the fatty acid tails of our phospholipid membranes are very hydrophobic, these guys can't just go through the membrane. They are blocked by those fatty acid tails. The only pathway that these hydrogen ions, these protons, have to get out of the lumen and back to the stroma is through the amazing ATP synthase. And so this movement of the hydrogen ions from their high concentration to their lower concentration through ATP synthase, this is coupled with that chemical reaction that binds together ADP and inorganic phosphate to make ATP. This is known as chemiosmosis, and it also happens in the mitochondria. We're going to make some ATP. This ATP is not like it's the same molecule. It is still ATP. It is just like ATP. However, its use is not the same use as the ATP that is being produced in mitochondria. That ATP is for growth and mitosis and producing hormones and producing proteins. This ATP is going to be used only for photosynthesis and the light-independent reactions that we're going to talk about in a couple slides. So this ATP, not regular usage ATP, that's the job of the mitochondria. This ATP is for glucose production only. Super cool, right? But wait, there's more. So let's go back to this cytochrome, the cytochrome that had these high energy electrons. That energy from the electrons was transferred to cytochrome so that the cytochrome could pump those hydrogen ions into the lumen of the thylakoid. So now these electrons are low energy electrons. Where are they going to go now? Well, they get picked up by yet another electron carrier molecule. This guy is called plastocyanin. The plastocyanin is going to carry these low energy electrons to the other photosystem that we haven't talked about yet. Photosystem number 1. Photosystem number 1, even though it's second in the story, we discovered it first, so it's photosystem 1. So photosystem 1 is going to grab onto those low energy electrons from the cytochrome complex, the electron transport chain. Photosystem 1 is going to also absorb more light energy. It also has those pigments. It also has a reaction center. It happens to be P700, so it absorbs, its peak absorbance is a slightly higher, longer wavelength. But it's pretty much the same idea, except there isn't an oxygen evolving complex. We're not going to have any more photolysis. We are going to re-excite these electrons. So now when P700 loses electrons, instead of passing them to the electron transport chain, they get passed instead to ferredoxin, which is then going to reduce. We're going to have some more reduction happening. We're going to reduce this molecule NADP to NADPH. So NADP, just like NAD in respiration, except there's this extra phosphate group on it. So we are going to grab onto some electrons and also some hydrogens to reduce the NADP to NADPH. Sometimes we simply call it reduced NADP, NADPH, same thing as reduced NAD. NADP. This NADPH, reduced NADP, is going to be used along with that ATP in the light independent reactions that we'll talk about in a couple slides. So we have been talking quite a bit about these protein complexes, the photosystems, electron transport chain, these electron carrier molecules, ATP synthase, and they are all bound in the thylakoid membranes. Here we're going to talk about some of the advantages of the specific structure of the thylakoids. So thylakoids are sometimes in these stacks. These are called the grana. Sometimes they are between the stacks, and this is known as the lamella. So what is a benefit of these stacks in the grana is that they have a very small space on the inside. So we have lots and lots of surface area, but then each one of these pancakes, the inside volume is pretty small. That small volume means that we can get a high concentration gradient, this proton gradient, built up pretty fast. We have lots of photolysis happening here inside the lumen of the thylakoid. That means that we have a constant supply of hydrogen ions and also electrons for our photosystem IIs. We also have our photosystems, our ATP synthase, the electron transport chain, those electron carrier molecules are all embedded pretty close to each other in the membranes, either in the grana or in the lamella. And having them close to each other means that it's pretty easy for us to transfer those electrons around the membranes, whether it's in the grana or in the lamella. Interestingly, photosystem II, the one that we talk about first, even though we discovered it second, where photolysis occurs, our P680, these are found more frequently in the thylakoid that makes up the grana. And then PS1, the photosystem that transfers electrons to NADP, that reduction stage, they're found more here in the lamella. And the reason that photosystem 1 is more efficient in the lamella is because there's just a lot more surface area with the stroma, and the stroma is where we're going to have those NADP molecules that are coming to get reduced with the electrons coming from photosystem 1. So let's kind of summarize, but also differentiate between photosystem 2 and photosystem 1. So photosystem 2 is what we talk about first. This is the guy that is going to first excite electrons. Those electrons will get passed over to the plastoquinone, to the cytochrome complex for the pumping of the hydrogen ions. So what I like about this diagram is it shows that excitement of the electrons as it gains energy from sunlight. Here we can see how the electrons are losing energy. They're kind of going back down as they're losing energy to the cytochrome, so the cytochrome can pump the hydrogen ions. And then we get back to photosystem 1, and that, again, re-excitement of those electrons. So now we have crazy, crazy high amounts of energy that is going to be used to reduce the NADP to NADPH. Where is that photosystem embedded in our thylakoids? It is more often in the grana. So photosystem 2, we're going to find more here in those stacks, whereas photosystem 1 is going to be more in the lamella, those spaces between the stacks of the thylakoid membranes. And the reason is just better surface area with the stroma to get to those NADP molecules. The reaction centers, photosystem 2, we're going to call it P680, and its job is to give electrons to that plastoquinone, which is part of the electron transport chain. The reaction center in photosystem 1 is P700, so it absorbs better a little longer wavelength of light, 700 nanometers. Its primary electron acceptor, who it's giving its electrons to, is going to be that NADP, which will then be reduced to NADPH. The source of electrons for photosystem 2, where does photosystem 2 get the electrons that it can then give to the electron transport chain? It is going to be from water, that photolysis of water that happens in the oxygen evolving complex, which is part of photosystem 2. Photosystem 1 is going to get electrons from the electron transport chain, specifically that plastocyanin that will take the low energy electrons from the cytochrome complex over to photosystem 1. The job of photosystem 2 is to pass electrons to cytochrome so that we can pump those protons for some lovely chemiosmosis, so that we can make lots and lots of ATP that we'll then use in the light-independent reactions that we're going to talk about on the very next slide. The job of PS1, photosystem 1, is to reduce that NADP to reduced NADP. We can also call it NADPH, which also is needed to make the glucose in the light-independent reactions. And finally, we're going to talk about those light-independent reactions. Here we go. So again, we made some ATP and we reduced some NADP, and this is going to be used in the light-independent reactions. It's known as Calvin cycle because it was deduced by an amazing scientist known as Melvin Calvin. Yes, his parents called him Melvin Calvin. He did some amazing stuff with some radioactive carbon, and we figured out these different stages of the Calvin cycle. So the first stage that we're going to talk about is here, and it is carbon fixation. So in sciences, this idea of fixing something just means to glue them together. So we have this enzyme Rubisco. Rubisco is going to glue together some carbon dioxide molecules to these guys, ribulose biphosphate. We sometimes call it RUBP for short. So ribulose biphosphate, RUBP, plus carbon dioxide is going to lead to the production of glycerate phosphate. We sometimes abbreviate that as GP. So we're going to take 3 CO2s and 1, 2, 3 RUBPs, and we end up making 6 molecules, because these guys kind of get bumped apart. We're going to make 6 molecules of this glycerate phosphate, or GP. These 6 GP glycerate 3-phosphate molecules are then going to be reduced. Reduction is our next stage in the Calvin cycle. We're going to reduce, remember Leo says Ger, and so, that's a G. Reduction is the gaining of electrons and also some hydrogen ions. So we're going to use some of that ATP that we made over here in the light-dependent reactions, and we're going to use some of that reduced NADP, also called NADPH, also made in the light-dependent reactions. We're going to use that ATP and that reduced NADP to reduce our GP. We're going to gain electrons and protons on this GP, and that's going to turn it into this other molecule called TP, triose phosphate. 6 total are made. One of them is going to exit the cycle, and it is going to become half a glucose molecule. The other 5 are then regenerated in the regeneration stage of the Calvin cycle, regenerated back into ribulose biphosphate using some more ATP. And so this ribulose biphosphate is going to again react with some more carbon dioxide using that enzyme Rubisco to make 6 GPs. That GP, glycerate 3-phosphate, is going to be reduced with ATP and NADPH to make some triose phosphate. One of them will exit and be the other half of a glucose molecule. The other 5 will be regenerated back into that RUBP so that we can do the cycle all over again. Notice that each turn made only half a glucose. So we have to do the Calvin cycle 1 time, 2 times in order to produce a whole entire glucose molecule. We're going to need 3 carbon dioxides for 1/2 of the glucose, another 3. So that's that total of 6 carbon dioxide molecules that are needed for the production of 1 glucose molecule. But wait, there's more. Most of the time when we talk about photosynthesis, we're focused just on that production of glucose. But also, we can take some of these GP molecules, these glycerate 3-phosphate molecules, and they can be used to make amino acids. The plants can actually make all 20 amino acids from glycerate 3-phosphate molecules to produce their own proteins. And then we go and eat them. And this is how us animals make our proteins. Some of these TP molecules can be used to make glucose. That glucose can then be stored as starch later on. But we can also use some of those triose phosphate molecules to make other lipids or nucleic acids, and then also our proteins. So some of these guys will exit the Calvin cycle and be used in other metabolic pathways to make all the other things that plants need to make. So even though we talk mostly about glucose, other products are possible. After the light-independent reactions, Calvin cycle, use the ATP to make the reduced GP-TP, or to turn the TP back into RUBP. That ATP is now converted back to ADP. That ADP then floats around the stroma until it runs into ATP synthase embedded in the thylakoid, and then it turns back into ATP. And that ATP is used, it converts back to ADP, and then it's turned back into ATP. So we can see this lovely interdependence between the light-independent reactions and the light-dependent reactions. Same thing happens with our NADP and our reduced NADP, NADPH. So we're going to take that NADP, it's going to gain some electrons and protons to turn into the reduced form, NADPH. That H gets lost when our GP is reduced to the TP, and then that NADP is going to be floating around the stroma of the chloroplast, and it's going to eventually run into photosystem 1, where it will be reduced back to NADPH so that it can do it all over again and all over again. So this interdependence leads to some rate limits for photosynthesis. So if we have a limit on carbon dioxide, if we have a very low concentration of carbon dioxide, then we're going to have a slow rate of photosynthesis. And the reason is because we don't have a lot of carbon fixation occurring. We need that CO2 here, the RUBP, being bound to the CO2 by that enzyme Rubisco, is going to happen here. Rubisco, as an enzyme, is actually pretty inefficient. It can only bind 3 carbon dioxide molecules per 1 second, which is really, really slow in terms of biochemical processes, which means that plants have a lot of Rubisco. We're pretty sure that Rubisco is the most common protein on the planet. We think that there's about 700 billion kilograms of it floating around on Earth. So that's a lot of just this one baby molecule Rubisco. So again, as we increase CO2 concentration, that Rubisco can start binding more CO2 to that RUBP, ribulose biphosphate, until eventually all of our Rubisco is working as fast as it can. And so we say here that the reaction has plateaued. The Rubisco is saturated. It's working as fast as it can. It cannot work any faster. Of course, if we added more Rubisco, we could speed things up. But if we're talking about a closed system where all the Rubisco is already there, we're going to plateau at a very high CO2 concentration. The other limiting factor that we're going to look at is light intensity. Here, we're talking more about those light-dependent reactions now. Very little light means that photosystem 2 and photosystem 1 are not absorbing energy, which means that we're not having that excitement of electrons. There's no excitement of electrons. There's no ATP production. There's no NADP reduction. And so therefore, not a whole lot of photosynthesis is going to occur. But as we increase the light intensity, more excitement of the electrons on the photosystems is going to occur until we have maxed out all of those photosystems. They're absorbing as much energy as they can. Any extra energy that runs into the photosystems will just be released as fluorescence. We talked about that when we talked about the flame test lab those many, many, many slides ago. And on that note, my friends, we have accomplished video lecture, additional higher level content on photosynthesis. We reviewed those basics of photosynthesis. We talked about that reaction. 6 carbon dioxide molecules and 12 waters are going to produce 1 glucose molecule and some oxygen molecules and some more water molecules. That CO2, carbon fixation in the light-independent reactions, all this water photolysis in the light-dependent reactions. That oxygen is produced in the oxygen-evolving complex of photosystem II. That glucose is produced when we have 2 triose phosphates produced in the Calvin cycle. We talked about the functions of photosystems 2 and 1. Oxygen production happens in the oxygen-evolving complex of photosystem II. ATP synthesis occurs due to the hydrogen ion concentration, that proton gradient, due to photosystem II working with cytochrome, and then also that NADP reduction due to photosystem number 1. We talked about the Calvin cycle. There are 3 main stages: carbon fixation, reduction, and then regeneration. And we talked about how that ATP that is made in the light-dependent reactions is used in the light-independent reactions, and then that ADP gets turned back into ATP in another round of light-dependent reactions. Same thing happens. We have some NADPH, that reduced form of NADP is produced in the light-dependent, which then is oxidized back to NADP in the light-independent reactions, but then it goes back to the light-dependent to get reduced. Great work today, my friends. Lots and lots of chemistry in this lecture. Good work.